Hi, I am Mrs. Sloan, and this is video number 2 of cellular respiration. This video is focusing on aerobic respiration. I want you to know my intended audience here is our AP biologists. The reason is I'm going to go into some regulating factors of it, and I'm going to get into how all of our biomolecules here in Unit 3, how this relates to fitness. So those are just some added things for the AP expectations. You're welcome to watch this video if you're in honors or CP bio. I will also be making a video for them as well. All right, so I want to remind you of what we already discussed in video 1. So we already went over the overview of cellular respiration. We talked about the different coenzymes that we are using here in cellular respiration. We also went into some detail about anaerobic versus aerobic respiration, and we focused on fermentation. And we talked about the three different ways of phosphorylation, of which the first two occur in these first two right here occur during cellular respiration. We also went over the steps of glycolysis. Remember, it is a 10-step process, but I'm going to simplify it here in the Minecraft version. Ultimately, in the 10 steps, glucose is broken in half. It's 10 different steps, 10 different enzymes. Your end product is you have these 2 pyruvic acids. Along the way to get your party started, you have to invest 2 ATP, but you get 4 ATP back. So that's why you net out 2 ATP here. Also, you reduce 2 NAD in the process. Remember, the whole point of fermentation, as we talked about that, was to oxidize your NAD that you had reduced during glycolysis so you could reuse it again here in glycolysis. All right. Now here we had no oxygen before. Now we do. Now we do have oxygen. So we want to talk about how this process is now going to be altered with the presence of oxygen. So here again is an overview. Glycolysis, 10 steps, right? 10 enzymes. You generate 2 ATP through substrate level phosphorylation, and you generate 2 reduced NAD. These are like checks, right? You can't go to a soda machine and put a check in there. You have to cash it out for money. The money we're trying to make is ATP. So these 2 NADs, instead of doing fermentation, we're going to send these to the electron transport chain in the mitochondria. The inner membrane of the mitochondria is where we're going to do that. That's called the cristae. And there, just like we did in photosynthesis, we're going to concentrate hydrogen ions, and then we're going to use that concentration of hydrogen ions so that the hydrogen ions can pass through ATP synthase complex and we can generate some ATP. The final electron acceptor is going to be oxygen, so it forms water. So glycolysis is still the same. Our second step in aerobic respiration, instead of fermentation, is we're going to do the transition or preparatory reaction, and this is where we convert the pyruvate into acetyl-CoA, which gets fed into the Krebs citric acid cycle or TCA cycle, which generates even more NAD and FAD that we can feed into our electron transport chain. All right, so let's look a little bit more on what the mitochondria actually looks like. And I'm going to go in presentation mode, and here I am again. All right, so when you look at the mitochondria, I want you to think a couple of things. It, like chloroplast, is an energy-related organelle. It, like chloroplast, was probably formed through the endosymbiotic hypothesis, where it was a free-living prokaryote that got engulfed and brought in. It has its own DNA. It's circular. It's self-replicating by binary fission. It has its own ribosomes. It can do protein synthesis, right? So remember, this inner membrane was probably the original membrane of the prokaryote, and this outer second membrane was a result of getting engulfed by another cell, right, to become its organelle. So on the anatomy, this interior compartment right here, the darkest color that you see right here, and in this bottom picture, it's purple, okay? This purpley stuff right here, that is referred to as the matrix, okay? That is the matrix. The part in this diagram that's this lighter pink right here, and which would be blue, and this is using an electron microscope to generate this image, is this blue part right there. That is referred to as the intermembrane space. Let me tell you something really important about this, okay? When we did photosynthesis, we concentrated the hydrogen ions to the centermost part of the chloroplast. Remember the green hollow pancakes? All the hydrogen ions were inside of them. And then when they went back out into the stroma, they generated ATP. This is opposite day. We're doing everything opposite of what we did in photosynthesis. So now in mitochondria, we are going to generate our hydrogen ions in this light pink, purple area called the inner membrane space. That's where our hydrogen ions are going to be concentrated. And they're going to go across this inner membrane, which is called the cristae right here, go across it into the matrix. And we're going to be generating the ATP in the center of the mitochondria, right? It's just opposite of what we learned in photosynthesis. Now, what kind of things occur also in the matrix? Well, when we take those 2 pyruvic acids that we made during glycolysis and we convert them to acetyl-CoA, those acetyl-CoA are going to be formed in the matrix. The Krebs citric acid cycle also is going to occur in the matrix. So on your notes, I am starting, and by the way, the notes are down in the descriptor of the video, okay, so you can have a copy of those. I am on 8.4. I did 8.1 through 8.3 in the first video, all right? 8.4 has structure reviewed. The inner membrane space is between the outer and the inner membrane. That's purple in the lower picture, light pink in the upper picture, okay? And this is the area where hydrogen ions are pumped or concentrated. The matrix is the location of the prep reaction or transition reaction. That's where we make acetyl-CoA and also the citric acid cycle. The cristae are the folds on the inner membrane, okay? These folds. Another question. Why all the folding? You know, you can see it in greater detail here. All this folding increases what? What are you going to tell me? Surface area, right? Surface area, good. So the cristae are the folds on the inner membrane, and it's the location of the electron transport chain. All right, so quick review of glycolysis, all right? One more time. You start out with glucose. You take those glucose molecule that has 6 carbons and you break it in half. 10 steps, 10 enzymes. You generate 2 pyruvic acids, each containing 3 carbons. Now, you lost some hydrogens along the way. You can see you went from H12 down here to 2 times 4, 8 hydrogens, right? That's because we reduced the NAD. So we transferred the high energy electrons from glucose into our mule NAD. Now he is holding those high energy electrons. Remember, you spend 2 ATP to get the party started. 4 ATP come as a result. So you generate 2 ATP. So now we have the 2 pyruvic acid. Now we're going to go into the next step, the preparatory or transition reaction. All right, I'm showing you, it says there's 2 of them, okay? But here is a pyruvic acid, the end product of glycolysis. Notice it has 3 carbons here, just like I told you, there's a three carbons, okay? And then what I want you to see right here is that we're adding in what is a coenzyme. Remember coenzymes, where that facilitates an enzyme so it can function better and it's organic? That's what CoA is. So to get it in the right spot, we're going to get this coenzyme and we're going to throw it right here on this pyruvic acid. But we need to make a spot for it to land. So what we're going to do is we're going to take these 3 carbons right here, and let me get a pointer real quick. Okay, we're going to take these 3 carbons right here, and we're going to take this carbon right here and remove it, okay? This carbon and these 2 oxygens. C and 2 oxygens. What is that? CO2, right? We're going to take this right here. That'll be CO2. Now breathe in, breathe out. Some of the CO2 you exhaled right now could have been from this reaction in your cells, all right? And we're going to put the CoA in its place. Now, the only thing we haven't dealt with is this hydrogen. I wonder where it's going. Do you see it? Do you see it? Yes, we're going to use it to reduce this NAD. So in the process of this transition reaction, we take these 2 pyruvic acids and we convert them to 2 acetyl-CoAs. I'm going from 3 carbon units to 2 carbon units. And I'm attaching a coenzyme on there that can be used in the next sets of reactions. And also, you reduce 2 NAD molecules. So what do we get when we walk away from this reaction? Well, to go on to the next step, we get 2 acetyl-CoAs. Also, these 2 reduced NADs can get sent to the electron transport chain and get cashed out for some ATP. So on preparatory reaction, it connects glycolysis to the citric acid cycle. And then I gave you all the notes for this. Each pyruvate is converted to acetyl-CoA. You remove a carbon, COOH, as CO2, and a hydrogen from each pyruvate. You use the electron from the hydrogen to reduce the NAD, which you can see up there on the top. And a molecule of CoA binds where the COOH once was located. So now we have pyruvic acid getting converted to 2 acetyl-CoAs. Each of these acetyl-CoAs is going to get fed into the next bit, the Krebs cycle. So if we have 2 acetyl-CoAs, how many turns of that cycle are we going to need to do? That would be 2. All right, let's go look at the Krebs cycle. This is an ugly diagram. It's from your book. I'm going to show you a better one in just a second, okay? I'm showing you this picture for a couple of things. Number one, you can see glycolysis kind of grayed out over here. Then you can see the preparatory reaction where acetyl-CoA is made that we're going to feed into our Krebs cycle. Then all the NADs and FADs that we make are all going to get fed into our electron transport chain. That's coming up, okay? Take a look down here at the citric acid cycle, Krebs cycle. Another name for it is TCA cycle. So it's all the same thing here on this cycle. Here's your acetyl-CoA. Remember, it has 2 carbons. We're going to hook it up with a molecule that has 4 carbons in it. What is 2 plus 4? It's 6, right? And so the first compound we make right here is citric acid. And another name for that is citrate. Look what happens. Acetyl-CoA, the CoA gets kicked off because we used it, okay? We're done with that. So that helped feed us in to this citric acid cycle. Now, as we go around, again, I'm going to show you a better picture. Just give me a minute. I wanted to show you this one first. Here's your citric acid. It has 6 carbons in it. You are going to reduce an NAD and pull off a CO2. You're going to do that simultaneously. This part right here is just like the preparatory reaction we did before. You're removing a carbon as carbon dioxide, and you're going to use some of the hydrogens, the bonds, the electrons in the citrate to reduce NAD. So you go from a 6-carbon molecule. When you remove a carbon dioxide, you're now down to a 5-carbon unit. Okay, this 5-carbon molecule, you are going to do the same thing. You are going to pull off a CO2 and reduce an NAD. Okay, so now we've gone from 5 down to 4, but you're also going to have the ability to make substrate level phosphorylation, make a single ATP molecule. You're also going to be able to pull some more electrons out to reduce the other coenzyme I mentioned, and that is FAD, and you're going to reduce that. Okay, it's still a 4-carbon unit. And look, you can pull off some more electrons still and make NAD. Now, when we made the NAD here, it wasn't like before because we didn't pull off any more CO2. Same thing here with the FAD, all right? So now we regenerate this 4-carbon unit. We can bring in our second acetyl-CoA and do the whole thing again, all right? Now, let me show you another diagram with that, okay? So here, let me move myself in here. Okay, so here you can see your acetyl-CoA. It's a 2-carbon unit. I'm going to remind you, there's hydrogens and oxygens there. I'm just following the money. I'm just following the carbons, okay? Preloaded in your Krebs cycle is this C4 molecule, okay? I'm going to add a 2-carbon unit to it, so now I make a C6 molecule, okay? Then I'm going to pull off a CO2. So I'm pulling off a carbon, and I'm going to reduce some NAD. I now have a 5-carbon unit, okay? Because I lost a carbon, right? Then I'm going to do that one more time. Pull off a CO2. I'm going to reduce an NAD. I have a 4-carbon unit. Then I'm going to make an ATP, substrate level phosphorylation, no electron transport chain needed. I'm going to reduce an FAD, and I'm going to reduce an NAD, and I'm going to do it all over again with my second acetyl-CoA. So once I've done this twice, right, I'm going to get 2 CO2s times 2 is 4 because I'm doing it twice. Plus I got 2 CO2s from the transition prep reaction. That explains my 6 CO2s overall. Every time I do the Krebs cycle, because I'm going to do this 2 times, every time I get 1, 2, 3 reduced NADs from the Krebs cycle. 2 times 3 is then 6 NADs total from this. I get 1 FAD times 2 is 2 FADs, right? And 2 ATPs made from it. That's just the math behind it. Okay, so take a look at your notes that I gave you. Okay, and I'm not going to read them all to you right now because you have access to them either through my class or through the YouTube down at the bottom in the descriptor. The citric acid Krebs cycle completes the oxidation of the original glucose. We've spent it. We have spent all the glucose at this point. Okay, I want to quickly remind you there are 6 carbons in glucose, right? We went from glucose and glycolysis to 2-pyruvic acid. I still have all 6 carbons there, right? In the transition reaction, I convert this 3-carbon pyruvate to a 2-carbon acetyl-CoA, and I give off a CO2 each time. So that's 2 CO2s right there. And then every time you do the Krebs cycle, you do 2 more. And since we do the Krebs cycle twice, that's 4. 2, 2, 2, that's 6. 6 carbons, they're all accounted for. So we have completed the oxidation of glucose. For each glucose molecule, the cycle occurs 2 times because we had 2 acetyl-CoAs. And then there's the song right there. And I'm going to teach you the song in just a minute. But that is for those of you who learn my songs, okay? Now, what we're ready to do now is to go ahead and cash out all these NADs and FADs for some ATP. So we're going to go to the, oh, I forgot I have this little summary. Pardon me. Okay, so here was the inputs for the citric acid cycle, 2 acetyl-CoAs, 6 oxidized NADs, 6 oxidized FADs. And then if I'm going to make ATP, I got to make it from something. So I make it from ADP. So these are our reactants of the citric acid cycle. Here are our products, right? 4 CO2s, 2 a turn, 3 NADs a turn. I do it twice, that's 6. 1 FAD a turn, I do it twice, that's 2. And each time I do it, I get 1 ATP, so a total of 2. So that's the math, all right? Now, let's go on to the electron transport chain. And I'm gonna move, go over here, good. Okay, so I'm gonna show you a couple of different pictures, but I want you to remember back to photosynthesis. Remember when we had the electrons now that we got from water. Do you remember that part? And we excited them with the sun and none. Now we're coming in with high energy electrons via NAD and FAD. Now, NAD made from the Krebs cycle, put a pin in the ones from glycolysis. I'll get back to that in a minute. NAD made from the Krebs cycle get to come in at the very tippy top of the chain. They get dropped off at the top of the chain. So they drop off their electrons, and now our NAD is oxidized, right? It had the electrons and it drops them off. Remember, all electron transport chains are a series of redox reactions, right? You're oxidizing the NAD and reducing the first molecule that accepts it. So these electrons are getting passed through the electron transport chain. Just you have them all over your cristae of your mitochondria, but when we do cellular respiration, it's just one chain that we have to worry about, right? Okay, but they're all these chains are all over the place in the mitochondria. So we're, instead of 2 photosystems, that's what I was referring to. So when you you have 3 different places where you can contribute to the hydrogen ion gradient. So you have one here, and it's taking hydrogen ions from the matrix to the inner membrane space. And then you have a second one here, and you have a third one here. So if you drop your electrons off at the top of the chain, you're sending 3 different places for hydrogen ions to go out. And we're going to say reductase, cytochrome oxidase in our song, these 3 right here. That means I have 3 hydrogen ions that can come in. So you would expect then 3 ATP to be made. Now, do all 3 ATP get made? No, because the second law of thermodynamics, right? Every transfer of energy, we lose a little bit. So they average out to about 2.5 ATP once you take into consideration that not all energy is transferred, all right? Those are NAD that come in at the top. They get to take advantage of all 3 of these transport molecules, and so they will make, 2.53 ATP. FAD is not as lucky. FAD is less powerful, so he doesn't get to come in at the very tippy top of the chain. He comes in partway through. And as a result of that, he misses the first hydrogen. okay? Because he jumps in a little bit lower, so he only pumps out when he drops off his electrons. They don't get to go na. They just get to go na. So as a result of that, you'd have to know my songs to know what that meant. They only pump out 2 hydrogens. That's all that they have, the power to move 2 hydrogens. Therefore, only 2 hydrogens come in, 2 ATP, technically about 1.5 when you take into consideration the second law of thermodynamics. All right. Keep in mind the gray here represents the cristae, the folds on the inner membrane of the mitochondria. And then this area right here would be the matrix. And this area out here would be the intermembrane space. Oh, which is labeled right here, which I forgot. All right. So if you look at the electron transport chain on your notes, the purpose is reduced NAD and FAD, bring electrons to the ETC, become oxidized, and return to the other steps of cellular respiration. So once this NAD drops off its electrons and the FAD drops off its electrons, it can now be reused in glycolysis, transition prep, or the Krebs cycle and get reused again and again. Now, I gave you some really detailed notes in the descriptor, so I'm going to encourage you to take a look at those. Now, I want to show you, talk about some NAD from glycolysis. Remember, transition prep is inside the mitochondria, and so is the citric acid cycle. Those NAD in there are the powerful ones that get to come in at the top. The 2 NAD that you made during glycolysis, that is out. Where does the glycolysis takes place? Out in the cytoplasm. And so those NADs, sometimes they will come all the way into the ETC, but sometimes what they'll do is they'll be like, I don't feel like it. And they'll drop off their electrons to FAD and have FAD be the mule to go into the electron transport chain inside the mitochondria. So some of those NAD from glycolysis won't always get you the 3 ATP. If they hand it off to FAD, then you will only get 2 ATP. All right, let's look at another picture, okay? Same thing, get your bearings, take a look around, okay? Now, let's look to see the intermembrane space is this dark orange out here. Take a look at this little mini-me mitochondria. See the dark orange up there? That's the intermembrane space where we're concentrating our hydrogen ions. The matrix of the mitochondria is this yellow color down here, which is yellow here in our diagram as well. Okay, here comes, look right here, glucose, and then it says, right, using the glucose, you're going to do glycolysis, transition prep, Krebs cycle, and you're going to generate these NAD. Notice that it says they're carrying electrons from food. Yeah, glucose or any other molecule we want to use right here. Okay, the NAD is dropping off the electrons and becoming oxidized so it can be reused again. Okay, now these electrons are going to get passed along at 3 places. We're going to call this first one right here, okay, reductase. This makes, okay, so reductase. The second one where it comes out right here, okay, number 3 is cytochrome reductase. So we're going to just say reductase. Okay, I'm just using the second word there. And then when it keeps getting passed along across that membrane, the last one right here is cytochrome oxidase. That's the third one where the hydrogen ions go out. Then they will come back in, each one of those, through, look right here, ATP synthase complex, generating ATP. So we are coupling, right? Let me make, I don't want to hit pause. Oh, here, sorry. Here I am. We are coupling 2 things together, right? We are coupling an electron transport chain, which is generating a hydrogen ion gradient for us, right? We're coupling that with the chemiosmotic synthesis of ATP, where hydrogen is allowed to travel from one side of the membrane to another side of the membrane. And remember, those hydrogens really want to come into the matrix. Why? Because there's a difference in charge, right? Because they're hydrogen ions, they're plus, right? So there's a difference of charge on either side of the membrane. There's a difference in concentration. There's more hydrogen ions on one side of the membrane than the other. And there's also a difference in pH because you have all those hydrogen ions out there in the intermembrane space and that lowers the pH. So there's 3 reasons why they want to come in. So the electron transport chain sets up the hydrogen and ion gradient. We couple that. Now we use that hydrogen ion gradient to help us generate ATP by allowing the hydrogen to come back in through ATP synthase complex. Okay, did that all make sense? Where is my pointer? Make myself a little bit smaller. Okay, now I just want to show you, because I like this picture a lot. I would screenshot this picture if I were you and put it in your notes. Okay, here is the picture that's in your book. I blew it up. It's a little bit blurry. Sorry about that. But there is a picture showing the same process in your book, so you can use that as well. Here, this is like a summary of all of aerobic respiration. So here you can see glycolysis up here at the top, right? Generating 2 ATP through substrate level phosphorylation. Then this pyruvate right here gets converted to acetyl-CoA in the transition prep reaction. This occurs now in the mitochondria. These acetyl-CoA go through the citric acid cycle, and each turn of the citric acid cycle makes 1 ATP, so you have a total of 2. These over here on the left, this is through substrate level phosphorylation. The rest of this ATP over here is using oxidative phosphorylation. You're using oxygen as your final electron acceptor. Oh my gosh, and I did not show you that. I am so sorry. Let me go back, okay? Sorry about that. So here, when you look at this, these electrons get passed along, If you want to keep this chain moving, right, you have an indoor of electrons from NAD, and you have an outdoor of electrons. You need to remove those electrons so you can keep, because these have a finite amount of electrons that each one of these molecules can hold, each of these protein complexes, that what they can hold. So our outdoor is at the bottom of the electron transport chain. Oxygen is the final electron acceptor, and it combines with hydrogens that are coming in, and it forms water. No oxygen, your electron transport chain backs up, your Krebs cycle backs up, and then you and I would die because we wouldn't have enough ATP, right? You can see that, right? If you don't have oxygen for like 2 minutes, you will pass out. The reason why you breathe is all that oxygen is the final electron acceptor at the bottom of all your electron transport chains and all of your mitochondria. That was key. Sorry, I forgot to mention that. Okay, so this all happens through oxidative phosphorylation along your electron transport chain. So this is just showing you the math. I had to do a correction because the book was wrong right here. So sorry about that. So the 2-NAD from glycolysis, remember I said sometimes they'll go in and then 2 times 3, right? If we're not talking about the second law of thermodynamics, 2 times 3 would be 6. But if it hands it off to FAD, then it would only get you 2 ATP. So that's why that says 4 to 6. The 2-NAD from transition prep, going from pyruvic acid to acetyl-CoA, each of those would get you potentially 3 ATP. So 2 times 3 is 6. 6 NAD from the 2 turns of the citric acid cycle would each potentially get you 3 ATP, so 6 times 3 is 18. And then the 2 FADs only would get you 2. 2 times 2 is 4. Again, this is the old school numbers, 36 to 38 ATP. It is not taking into consideration the second law of thermodynamics. Again, the anatomy is in your notes. The ATP production is in your notes, and talking about oxygen as the final electron acceptor is also in your notes, as well as the energy yield, the overall energy yield. Now, I have a couple summary diagrams that I found that I thought were kind of cool here that might be helpful for you. Look at glucose coming into your cell, right? This blue right here would be your cell membrane or your plasma membrane, right? Glucose comes in via a SIM port, so it has to kind of get escorted into the cell. Here's your glycolysis. Let's look at all of our options from glycolysis, right? We know as a result of glycolysis, you make 2 pyruvates right here. You know you reduce 2 NADs. Now, if you do not have any oxygen, then those NADs are going to get oxidized through the process of fermentation. And in you and I, in animal cells, the byproduct of that will be lactate, okay? Now, lactate gets removed. there's another whole thing we could talk about that with your liver. That's another day. But ultimately, you wait. This lactate can make your muscles sore, and so you want to oxidize it back into pyruvic acid when you and I have enough oxygen to take soreness out of our muscle, and then go the oxygen route. All right. Now, if oxygen is present, then what you're going to do is you're going to take this pyruvate and you're going to convert it to acetyl-CoA. And so here you can see pyruvate getting converted to acetyl-CoA in the matrix of the mitochondria. Look at this. We're reducing some more NAD and we're pulling off CO2. Notice they're showing you just one pyruvate, right? If they showed you the 2 pyruvates we have, you would see 2 CO2 here, and you would see 2 reduced NAD, and you would see 2 acetyl-CoA. Okay, I just want to follow you. They're just showing you one pyruvate through this one. Here is the TCA cycle, another name for the Krebs cycle. And when the acetyl-CoA gets fed into that, you get reduced NAD and reduced FAD. So these reduced NADs and FADs are going to go to your electron transport chain, which is going to generate all of this ATP that you see right here. This ATP right here came from glycolysis. And a byproduct of both the transition reaction and the TCA cycle is carbon dioxide and water. Okay, here is another summary diagram. Okay, you can see now you're inside the cell. You're not seeing the cell membrane. You're inside the cell and you're doing glycolysis. Again, that takes place out in the cytoplasm. And here you can see glucose going to pyruvate. You're generating net total of 2 ATP. Okay, and you form 2 reduced NAD. This is making the assumption that you have oxygen. They're not showing you fermentation in this picture. And the pyruvate is moving to the interior, right? Crossing the outer membrane and the inner membrane of the mighty mitochondria. And the pyruvate is getting converted to acetyl-CoA, again, generating CO2 and reduced NAD. This acetyl-CoA is going to be put into the Krebs cycle. I put down here for you, TCA cycle is the same as the Krebs citric acid cycle. It stands for tricarboxylic acid cycle, just another name, okay? You're going to do this. You're going to generate, and again, it's showing you one turn, right? As if you had one pyruvate molecule, but you will have 2 from a glucose molecule, okay? So it's showing you the 2 CO2 here. It's showing you that you make ATP, and it's showing more reduced NAD and reduced FAD. These reduced NADs and FADs, okay, are getting brought to your electron transport chain. They're going through the electron transport chain, and hydrogen ions are put into the inner membrane space, right? And then they're going to want to go from a higher concentration to a lower concentration back into the matrix, which generates ATP. Okay. Now, if you look up here, do you see where it says triglycerides? That's just another name for fat, right? A type of fat. Remember, it's glycerol with the long fatty acid chains. Those long fatty acid chains can be snipped into groups of 2, and those fatty acids can then be brought in right at the acetyl-CoA point. Now, what has more calories? Carbs or fats? What has more calories? Hopefully you're telling me fats, right? If you ever kind of follow nutritional information, fats have more calories, and this is why. Ready? Glucose gets broken in half. You have 2 pyruvic acids, and each of those get converted to 2 acetyl-CoAs, 2 acetyl-CoAs total. Fats, if you recall, 1 glycerol and 3 long fatty acid chains that could be anywhere between 16 and 18 carbons long. Each of those fatty acid chains, let's say it's 18 carbons long. If you snip it into groups of 2, that would be 9 acetyl-CoAs per fat, right? Per fatty acid chain. You have 3 fatty acid chains, 9, 9, 9. 3 times 9 is 27, okay? A single fat will generate 27 acetyl-CoAs, whereas a single glucose molecule will only generate 2 acetyl-CoAs, 2 acetyl-CoAs. All right, so that's where they would come in. Now I have another picture. I'm going to show you here. Okay, so on your notes, let me show you where I am on the notes. I have now jumped to metabolism 8.5. Okay. And remember, metabolism is all the enzyme mediated reactions of your body. Okay. So there are those that break down. That's a catabolic reaction. And then those that build, those are anabolic. So what this is just showing you is catabolic pathways release energy. The oxidation of glucose releases energy. And you can use that energy for anabolic pathways for building, just like you could build ATP, or you could use that energy from the ATP to build muscles. That would be an anabolic pathway. So on your notes, catabolism is degradative. It tends to be exergonic, so it's releasing energy. And in cellular respiration, besides glucose, let me show you, I got to get on another picture here. Okay, besides glucose, so here's carbohydrate. Oh, I think this is stir fry up here. I don't know. But you can use carbohydrates, glucose, glycolysis. This is the way we know. Okay, we know this way. Okay, fats I already talked to you about. You can break it into glycerol, which can go in as, right, it's 3 carbons, so it can go in as pyruvate. And then all the fatty acids can be snipped into acetyl-CoAs and go in right here. Now let's talk about proteins. Proteins right here are built out of amino acids. Now, what has to happen first is deamination, and that's going to occur in the liver, and that's when you remove the amino group. Remember, amino acids have a central carbon. You have the N-terminus in, the amino group, the carboxyl group. You have an R group, and you have an H, right? So you'll remove the amino group, and guess what? That gets converted to urea, which you pee out. Okay, the rest of the amino acid can either go in as pyruvate, acetyl-CoA, or into the citric acid cycle. So this is how we use all of those nutrients. All right. So anabolism. Oh, sorry. Go back into your notes. Catabolism on 3A. Proteins come in at all three steps. And fats come in as pyruvate. And that would be the glycerol, and by snipping the long chains into acetyl-CoAs. And then anabolism, you already know, ATP produced during catabolic reactions drive endergonic anabolic reactions. Okay, we're so close. Okay, so next, let's compare, okay, there. Let's compare chloroplast with mitochondria. Okay, so this is just a little simple diagram to do that. On your notes, I have the similarities there. They both use a membrane. For the chloroplast, remember it's a thylakoid membranes right here, the inner membranes here. For mitochondria, they're using the cristae right here, right? They both use membranes. They both have electron transport chains across those membranes to establish an electrochemical gradient. And they synthesize ATP through chemiosmosis. And they both use cycles in their enzymes, right? Because you have the Krebs cycle here and you have the Calvin cycle over here, right? So that's all in your notes. And then the big picture right here I want to talk about is chloroplasts are only found in plants, right? Because they're able to harvest light energy and transfer that into the chemical energy of ATP and reduced NADP in order to fix CO2 into sugars, right? So the flow of the energy is going to start from the sun. It's going to go into the chloroplast, and then you use mitochondria. And both plant and animal cells have mitochondria. All eukaryotic cells, right, are going to have mitochondria, and they're going to oxidize that glucose to generate a tremendous amount of ATP. So mitochondria don't need sunlight, but chloroplasts do. Okay, here is another one. Not scary, I promise. Just take a minute to take a look at it. Just absorb it for a minute. Okay. If you look over here, you have a mitochondria, a single mitochondria. Notice this is what we just talked about, mitochondria. Remember, the hydrogen ions get concentrated out in the inner membrane space, and then they come back in, right? Here it is coming back in through ATP synthase complex to generate ATP. Contrast that right here to your chloroplast. Remember, we concentrated the hydrogen ions inside the thylakoid membranes, and then when they went back out into the stroma, that's where we generated our ATP. Also, you can see light energy is what's exciting the electrons for the electron transport chain, whereas here for our electron transport chain, we're getting reduced substances, NAD and FAD, right? Now, notice here in the mitochondria, our final electron acceptor is oxygen. That's why you and I need oxygen as the final electron acceptor. and it forms water. So the irony here is the final product of mitochondria, right, is oxygen forming water. But in the chloroplast, what was the first thing we did? We split water, right? We split water and we used those electrons, right, to go through our photosystems, and we released oxygen. So just kind of comparing and contrasting those two. All right, now, what is this thing? This is fructose 6-phosphate right here at phosphofructokinase. That's what this whole thing is called right here. And this is a regulator, okay? And let me explain. If you look in here where it says fructose 6-phosphate and fructose 1,6-biphosphate, that is one of the steps. Remember I told you in glycolysis there were 10 steps? This is one of the 10 steps in glycolysis, and this would be feedback inhibition. So in cellular respiration, right, our whole point is to generate a lot of ATP. And so when you have enough ATP, when ATP binds here, it deactivates this enzyme, so you can't do one of your steps in glycolysis. But that's okay, we don't need to because we have plenty of ATP. So now we don't need to oxidize that glucose. What will we do with that excess glucose? I know, we'll probably store it as fat somewhere in our body, right? Because you need any more ATP. But as you do more things and you use up your stored ATP, then it will not be able to give that inhibition on the phosphofructokinase right here. And as a result of that, then you will do glycolysis again and continue on in those reactions. Okay, so on your notes, it says, excellent example of negative feedback. Phosphofructokinase is an allosteric enzyme active early in the pathway of glycolysis. It is inhibited by high levels of ATP. Thus, in turn, it stops the catalytic pathway of glycolysis. If you stop glycolysis, you stop transition, Krebs, and the electron transport chain. PFK, phosphofructokinase, which is this big green thing you're looking at right here, is considered a pacemaker of respiration. Okay, you are doing awesome. Hang in there. If you need to take a stretch break, do. This is all about AP biology right now, this next one too, because at the very end of this unit, which is what we're coming to, at the very end of this unit, then they take a moment to address fitness. Now, remember what we said, what fitness is, right? We introduced that in chapter 1. Fitness is not how many push-ups, sit-ups, what your heart rate is. Fitness is your ability to pass on your genes. And what they want you to see right here is that all of these biomolecules and the variety of biomolecules that we learned about, the organic molecules we learned about earlier, the carbs, proteins, lipids, nucleic acids, and all the variety of chlorophyll that we learned about, and the variety of lipids that we learned about, these things increase our fitness. So this is the final thing you need to learn for Unit 3 for APBio. Okay, so hang in there. You're doing awesome. I'm already proud of you. So cellular fitness, this individual is obvious, looks fit to me, but cellular fitness is not talking about the body, but it's talking about the variety of molecules that we have. So here, okay, here are the 4 important organic molecules. This is a great review for you, right? Amino acids build proteins, fatty acids can build, with glycerol, build fats, and you can build phospholipids. Carbohydrates, right? You have monosaccharides, polysaccharides, right? Remember, polysaccharides have they're either structurally important or we use them to store energy, but long-term energy would be fats, obviously. And then we have nucleotides are going to build DNA, RNA, and ATP. So on your notes, underneath fitness, okay, and this is referring in AP, if you look at the expectations, this is 3.7. Naturally occurring diversity among and between components within biological systems affects interactions within the environment. So variation at the molecular level. So here's one example. At the molecular level, it gives organisms the ability to respond to a variety of environmental stimuli. So having a variety of molecules that make up our cells, right, gives an organism the ability to respond to environmental stimuli. The number and types of molecules within cells provides organisms with a greater ability to survive and reproduce in different environments, right? Because we can be in different environments, that means we can survive in different environments, which means we can reproduce in different environments, which increases our fitness, okay? Increases our fitness. All right, let me give you some more examples, all right? So variety allows for better adaptation to a cell's environment or stages of development. So here's one type of variety, okay? Variety in phospholipids. So on this one, depending on the environment and what is needed for temperature variations. So you don't need to memorize all of these, all right? But what you're seeing right here is that they're just minor modifications in these phospholipids that help us to adapt to different climates, all right? So that's number one. The second one I want to show you is variety in hemoglobin. Okay, so hemoglobin is found within red blood cells, and you have about 250 million molecules of hemoglobin inside every single one of your red blood cells. Now, hemoglobin carries oxygen. If you have a body that can become pregnant, okay, your embryo within your uterus is going to need oxygen, right? But if your hemoglobin is hanging on to the oxygen, how would the fetus ever get it, right? Because we would release the oxygen to our tissues in our body, okay? But the fetus would be getting not enough oxygen for what it needs. So what's interesting is the hemoglobin you have now is different than when you were in a uterus. The hemoglobin you have now is different than what you had in a uterus. This hemoglobin here in this person, right? This is fetal hemoglobin in the blue line right here, where we would tend to release oxygen to our tissues right here if you look at 0.5, let's just follow 0.5 across right here. When we would release it at this saturation point, our hemoglobin currently, fetal hemoglobin is still loading at that point, and that's good. So we will release it. The fetal hemoglobin would still hang on to it. If these lines were exactly the same, if they were on top of it, the fetus would not be able to hang on to any oxygen because it would unload it at the same way that our hemoglobin currently unloads. So that changes during development for us. And this facilitates the flow of oxygen from the mother's blood to the baby's blood so it can carry it around to all of its tissues and drop it off at its tissues. So your hemoglobin varies during our stages of development. So if you look at hemoglobin F as a fetus, will pull oxygen and hang on to it. It has increased stickiness when the environment is relatively low in oxygen. And that oxygen is coming from the mother's blood. So hemoglobin A, after we are born, we don't want that stickiness anymore because we want to release that oxygen to our tissues so our hemoglobin changes because it would be difficult to get the oxygen to the various tissues that need it. Difficult to get the oxygen to the various tissues that need it. All right, and your last illustrative example is going to be with chlorophyll. Remember when we learned in photosynthesis that there were different chlorophylls? So this one right here is chlorophyll A, chlorophyll B. You remember there was carotenoids, and I told you xanthophylls. So by having a variety of pigments, you can absorb different wavelengths of light, which makes that plant more likely to survive and reproduce. All right? That is the end. Awesome. Great job. I'm super proud of you. Review all of Unit 3. All right? Yay.